Hey everybody, in this video we are going to cover DNA methylation, specifically in eukaryotes. So DNA methylation also occurs in prokaryotes, but in this video we are going to talk about it with respect to eukaryotes. And so also we are going to specifically talk about methylation of cytosine. So I know we don't need to memorize the structures of the different nitrogenous bases in this course, but here is the structure of cytosine, and this is where it connects to the 5-carbon sugar. Now, when cytosine is methylated by an enzyme called a DNA methyltransferase, it will have the following structure. So overall, the structure is identical, or nearly identical. The double bonds in the same locations. And the 5-carbon sugar bound to the same nitrogen. And the methyl group is going to go right here. And this, if this is cytosine, this right here, this is 5-methyl cytosine. Because the numbering goes like this: 1, 2, 3, 4, 5. So the methyl group is on the 5th atom of this molecule, or it's at the 5 position. Now, okay, so 5-methyl cytosine is often methylated in eukaryotes when, I should say cytosine is often methylated in eukaryotes when it is part of a CpG dinucleotide. So if this is a strand of DNA, this is the complementary strand down here. We have this P in here. So what does that P mean? Well, let's say this is, I'm just going to copy this down here. Now, CpG, this is the 5' nucleotide. This is the 3' nucleotide, and this P is just used to indicate that phosphodiester linkage, right? Phosphodiester linkage. And why do we bother saying CpG? Well, the reason is if we were to just say CG, well, then we might be confused. Is this geneticist talking about this CG, the hydrogen bonding that occurs between a C when it's paired with this G down here in the opposite strand? Or is this geneticist talking about the G that is adjacent to a C, on a single strand that occurs, sometimes? So CpG refers to a C linked to another G through a phosphodiester linkage. So these are on the same strand, and that's why we put that P in there. So the cytosines are often methylated when they are found in these CpG dinucleotides in DNA. Now, mammalian genomes have these things that are rather interesting, and they are called CpG islands. Now, CpG islands are these areas where there are an unusually high number of CpG dinucleotides. Now, they are about 300 to 3,000 base pairs long. There are many of these, and they are found in or near 40% of known promoters. So we think they're important with respect to transcription. Now, we also think that, or the evidence seems to suggest that DNA methylation typically inhibits transcription. So why would DNA methylation, cytosine methylation, inhibit transcription? Or at least what is the evidence for that? So evidence that DNA methylation inhibits, now typically, I should say typically inhibits, because this is biology and there are always outliers or things that go against the typical phenomenon. Evidence that DNA methylation typically inhibits transcription comes from findings that, CpG islands in promoters of what we're going to call housekeeping genes. Now, this is a new term for this course. So what is a housekeeping gene? A housekeeping gene is a gene that should be expressed all the time for that cell to be viable, or be, it's like what would be an example? Say a gene that goes an enzyme needed to use glucose or make ATP, something that the cell always needs to be transcribed in order for that cell to remain viable, to survive. So that would be a housekeeping gene. So CpG islands in promoters of housekeeping genes almost never methylated. So, okay, so that means that, these genes are always transcribed and these are almost never methylated. So it suggests that if they were methylated, well, maybe these couldn't be transcribed. But importantly, what goes with this first finding is that CpG islands in promoters of non-housekeeping genes, so these are genes that don't need to be on in every cell, CpG islands in promoters of non-housekeeping genes tend to be methylated in cells that do not express those genes. Okay, so we have a nice correlation there between CpG island methylation and transcription of genes. So evidence suggests that, DNA methylation inhibits transcription, and how? How might it do this? So some of the ideas are that methylation, let's say, how does it block transcription? So DNA methylation may, let's do it like that, DNA methylation may, number one, block binding of transcriptional activators, or general transcription factors to DNA sequences. Another possibility, DNA methylation may attract methyl, it's a methyl group, binding proteins. And some of these methyl binding proteins may inhibit transcription. Some of these methyl binding proteins that bind methylated DNA are known to attract histone tail modifying enzymes. So, and those histone tail modifying enzymes may modify the histone tails in a way that prevents transcription of the nearby genes. So, we have two ways that DNA methylation may inhibit transcription there. And again, these are very active areas of research. So another interesting thing about DNA methylation is that DNA methylation is heritable. So a process called maintenance methylation allows DNA methylation to be heritable. So let's take a sequence here, an example sequence of DNA, and I'll put maybe 2 A's there, 2 T's here. Let's say this is the 5' end, 3', 5', 3', and then here will be our CpG dinucleotide. I won't put the P in there. The P will be right here. Maybe I will put the P in there. And I'll put 2 more A's over here. Now, if this is methylated, this sequence is methylated, full methylation means this C here is methylated and that C down there is methylated. Both of these are 5-methyl cytosines. And this DNA molecule would be fully methylated. Now, after DNA replication, what happens is, this is the template for one new molecule, and this one down here is the template for the other new molecule. So we end up with hemimethylated DNA molecules. So let's say this strand is the template. It went up here. So that's methylated, right? That cytosine was methylated. And 5-methyl cytosine still pairs with G, so no problems replicating these. And here, this cytosine is not methylated. So this is hemimethylated. Now, same thing down here, except the opposite strand was the template. And that one is methylated. There's a G here. And I could put the 5' and 3' in. Okay, again, another hemimethylated molecule down here, hemimethylated. So we're missing this. So this cytosine down here is not methylated, and this one right here is not methylated. So we have 2 hemimethylated molecules. And after DNA replication, there's a DNA methyltransferase that scans the newly replicated DNA molecules and looks for hemimethylated DNA sequences. And when it finds it, it methylates the non-methylated cytosine and turns the hemimethylated sequences into fully methylated sequences. So I can put that in here. So there's the new methyl group there, and there's the other one over here. Okay, so that's why DNA methylation is heritable. The fully methylated sequences become hemimethylated after replication, but then a DNA methyltransferase comes in, looks for the hemimethylated sequences, and then turns them into fully methylated sequences. So once methylated, a DNA molecule tends to stay methylated by maintenance methylation. So we have a nice example coming up in the next video of how DNA methylation, it's hard to read that, I'm sure, but DNA methylation controls a really neat epigenetic phenomenon. I'll discuss this term also in the next video. Epigenetic phenomenon called paternal or maternal, depending on which example I use, paternal or maternal inheritance. So really neat phenomenon controlled by DNA methylation, and we're going to talk about this phenomenon in the next video. Okay, see you then.